It's my pleasure to introduce Dr. Angela Rasmussen. Angela grew up in Washington. She went to Smith College and got her Ph.D. from Columbia. She then returned to the Pacific Northwest for a postdoctoral fellowship, and she's a member of the microbiology department. Her work is on the Ebola virus, another very important emerging infectious agent that you probably read a lot about last year. And let me also comment that Angela is a huge Seahawks fan. She has 2 senior rescue pugs. I almost thought that was pigs, but fortunately I looked carefully, it's pugs.
It's hard to tell the difference.
And she was a contestant on the game Jeopardy! So please welcome Dr. Rasmussen.
Thank you.
You're welcome.
Well, thank you so much for having me here. This is really an honor to talk to the mini med school crowd. And hopefully today, I will be able to tell you some things about emerging viruses, which are also parasites. And hopefully, I think it will be very synergistic with the talk that we just heard. So I'm going to talk a little bit about other emerging viruses, but I'm mostly going to focus on Ebola because that was sort of the flavor of the year, virus-wise, last year. Of course, now everybody's talking about Zika, so I'll get to that at the end of the talk. But really, I'm going to use the West African Ebola outbreak and sort of the history of Ebola outbreaks to sort of explain what emerging viruses are, how they emerge, how they cause disease, how these things are transmitted, how they spill over from their natural reservoirs into human populations, what we can learn from these epidemics of emerging diseases, and how we're going to study them going forward. So first, I'll tell you a little bit about Ebola, and this is a story that many of you who've read The Hot Zone may be somewhat familiar with. Ebola is a relatively new virus in terms of when it emerged. It emerged in 1976. But there are a number of factors that go into the emergence of any virus. And so I'm just going to point a few of these out really quickly before I get started. One, and we heard a lot about this during the Ebola outbreak, is that global travel is a great way to take one virus from one part of the world and introduce it into a naive population in another part of the world. And that's been happening since, you know, the time of the conquistadors, when South Americans were decimated by smallpox that they had never seen before, when the Spanish explorers, conquistadors, invaded their areas. Another major issue is development. So the world population is increasing. We need new places to live. We're encroaching into these wild spaces where people have not previously been. And in the course of that, we're encountering pathogens, in some cases viruses. This is made worse by climate change. So we just heard a lot about a parasite that's transmitted by mosquitoes. There are a lot of viruses that are also parasites that are also transmitted by mosquitoes, such as dengue virus and now Zika virus. The mosquitoes that transmit these are moving into new geographic areas because climate change is making it possible for them to find new places to live. Resource extraction, so mineral resources, mining, oil drilling, fracking, those sorts of things where you're getting into caves and cave-dwelling animals that are not commonly encountering humans. You can spill over viruses from that. There's emerging comorbidities such as diabetes, which in some cases appear to make certain viral infections worse. MERS coronavirus is one example of a virus that seems to be associated with diabetes in terms of the most severe cases. There's water supplies, issues with access to food, and lest you think this is a problem that is restricted to the developing world, this picture right here, sorry, I guess I don't have a pointer. This picture of this water supply situation is in Flint, Michigan from about 3 weeks ago. So water supplies are a major public health risk all around the world. Eating wild game, foraging for wild animals in the forest, hunting, getting animals, getting exposed to animals. This is a date palm sap collecting bucket. This is how they think that Nipah virus is transmitted in Bangladesh. The humans put these date palm sap collectors at the top of these date palms. They collect the sap, and it's a refreshing local beverage. Also, bats fly around in these trees, and they urinate and defecate into these date sap pots, and people that way are exposed to Nipah virus and potentially other pathogens. There are many pathogens that are primarily agricultural pathogens, but can also affect human populations. Rift Valley fever virus is a great example of that. It causes terrible problems with, they call them abortion storms. Basically, it can devastate a region economically when all the livestock are unable to carry pregnancies to term, but that can be transmitted as well, and it causes a hemorrhagic fever in many of the people who are exposed to that livestock. It's also transmitted by mosquitoes. And then finally, cultural practices. So one thing I will talk about in a little bit in this talk is that one of the earliest risk factors for contracting severe Ebola is your involvement in burial, a traditional burial, which is usually done by the women in many of these communities. They would wash the family member's body, take care of all these traditional rituals, and in the course of that, expose them to huge levels of virus. So all of these factors can contribute to virus emergence. Many of them can work in concert to result in these viruses that we've never heard of, like Zika, coming out of seemingly nowhere and causing these huge epidemics. So back to Ebola and Marburg. These are a family of viruses called filoviruses, and they were very cleverly named because now this iconic Ebola morphology, they look like a thread. And that's what philo means. It means it's derived from Latin for phyllis, which means a thread. So thread-looking virus. They're what are called negative sense RNA viruses. So we have DNA for our genomes. These have RNA. What that means effectively is that they can undergo more mutation than organisms with DNA genomes. They cause a broad range of disease. So one myth that I would like to counter or rebuke is that everybody who gets Ebola is, like a fountain of blood, like something out of a horror movie. There are many people who get very severe Ebola disease who never have any hemorrhagic symptoms at all, which is actually why the World Health Organization has now stopped calling Ebola hemorrhagic fever, and they've started calling it Ebola virus disease to really reflect the true range of disease presentations of this virus. There are also suspected people who have mild or asymptomatic infections. And this is a little harder to understand because obviously if you're not sick, you're not going to go to a hospital and be diagnosed with Ebola. But there are people who have antibodies to Ebola suggesting that they've been exposed at least to something that looks a lot like Ebola to the immune system, and it has not caused an Ebola-like disease in them. So human outbreaks can, to either Ebola or Marburg viruses can be caused either by exposure to their natural reservoir, which is thought to be bats, so mining, spelunking, cave-type activities, hanging out in trees where bats roost is actually how they think the 2014 West African outbreak started. Spillover from non-human primates in the wild. So people are out hunting, they find a dead monkey, they find a sick monkey, they kill a monkey, they bring it back, they butcher it, cook it, touch it, any of that stuff, you can get a pretty severe dose of Ebola from handling an infected non-human primate. Most non-human primates are highly susceptible to the same type of disease that humans get with Ebola. And then finally, in a few cases, there have been infections caused by exposure to research animals. That has not happened very often, fortunately. It's only happened about twice. But there's the two major branches of the Filoviridae, which is the name of the filovirus family, are Marburg and Ebola. And really the first one that ever appeared was Marburg. Viruses are traditionally named after where they're discovered. And you might say, Marburg sounds like it's in Germany, which it is. These monkeys were actually from Uganda, so they were from Central Africa. But what happened was the way that they used to make polio vaccines was that they would get monkeys imported from Africa. They would dissect out the kidneys and use those kidneys to grow cells in culture, and they would grow polio vaccines in those. So these animals were being processed for use in manufacturing polio vaccines, and all of the workers started getting what they thought at the time was yellow fever. And it turned out, when they looked at it under an electron microscope, it had this unusual morphology, and it was like nothing anybody had ever seen before. It also was associated with much more severe hemorrhaging than you normally see with yellow fever. So this was really the first time a filovirus emerged. It was named MARBURG. Now there's currently a large, highly pathogenic research facility in Marburg where they continue to do a lot of work on Marburg and Ebola viruses. But this was really the first time that we ever saw a filovirus or saw these severe viral hemorrhagic fevers caused so consistently by one virus. And the reason why this emerged was, as I mentioned before, inadequate biocontainment, exposure to infected non-human primate tissues. Marburg emerged again. There have been several sporadic Marburg outbreaks in the intervening time, but Marburg emerged again with a vengeance in 2005. And the reason for this emergence was unknown, but what's notable about this is that unlike the previous Marburg outbreak, which had about a 30% mortality rate, this one looked a lot more like Zaire Ebola virus, 90% mortality, but it spread to all these 252 people, largely because of poor healthcare infrastructure and a delayed public health response. And that's going to be a theme that I'm going to discuss more, because there's only so much that we can do about some things like climate change with regards to virus emergence, but there are many things that we can do, and we need to learn these lessons from these past outbreaks. So Marburg tends to be associated a lot with mines and caves, and there was another outbreak. This was only 3 cases in 3 gold miners, and they actually went into this mine, officials from the World Health Organization went into this mine, trapped a bunch of these Rosetus aegypticus bats, and found Marburg RNA in some of these. So they discovered then that the bats are probably the reservoirs for these filoviruses in the wild, which was a question that people had been trying to answer for a very long time, since Marburg first emerged really in the 60s. So this was another example of people going into a cave and getting it from the bats. There was another situation where this cave is part of a national park in Uganda. There were 2 cases. One was a lady who was, I believe, from the Netherlands. She died. The other was an adventure travel writer who went back to Colorado, got sick, was mildly ill, ended up going to the hospital. She got more severely ill. They couldn't figure out what was wrong with her. They tested her for everything, and finally they tested her for Marburg. She was seropositive, or she had antibodies in her blood for Marburg, and they realized after the fact that she probably got Marburg when she was exploring in this cave, which literally has like 5 million bats living in it. I've seen pictures of it. It's insane. And there's also big snakes. It's named Python Cave because there's pythons and forest cobras that live in this cave. It doesn't really seem like an awesome place to go visit necessarily, but there have been at least 2 cases of Marburg that have come out of there. So we started through all of these outbreaks over the years to understand a little bit more about Marburg virus, and we still don't understand entirely how this relates to Ebola. Marburg and Ebola have many things in common. They're cousins, essentially, taxonomically, but they do have many differences. And in fact, even some of their viral proteins, which we thought did the same thing, actually don't, and they work very differently in Ebola versus Marburg. But Ebola emerged in 1976 in Yambuku, Zaire. Now it's the Democratic Republic of Congo. The reason why this emerged is unknown, but how it spread is very well documented. And that was, there was a clinic run by these nuns, and they would give people their anti-malarial shots. And they had like 4 or 5 syringes. And basically what they would do is they would pull up a big dose of these anti-malarials. People would line up outside, and they just give them the shots, one after the other. So this was before the era of HIV. You know, we didn't understand about HIV, HBV, all these bloodborne illnesses that we're now very aware of and hyper-vigilant about in terms of medical care. At the time, it was completely normal, especially in these resource-poor settings. It was in a very remote village, to reuse medical sharps, and that was how it spread through this community. Furthermore, there was a big highway that was basically developed at the time that connected this town to Kinshasa, which is the capital, major city. So people started going to Kinshasa, and that was where they began infecting healthcare workers. And Nurse Mainga, who, I don't have a picture of Nurse Mainga here actually, but Mainga is the name of the prototypic strain of Zaire Ebola virus that we work with in the lab, and it's named after this unfortunate woman who was a nurse, treated these patients when they got to the hospital in Kinshasa, and then contracted Ebola herself and died. It's a very sad story, and again, as with many of these outbreaks, a more rapid response and a better understanding of what we were dealing with at the time of emergence could have saved lives and changed the outcome of this experience. But this index outbreak, or this initial outbreak of Ebola, was followed very closely after by another outbreak of another Ebola virus called Sudan virus, or Sudan Ebola virus, in Sudan, right around the same time. Why these two different species of Ebola emerged at the same time, we have no idea, and we still don't really have a good understanding of why these things just pop up out of nowhere. But this outbreak is where you always get that, the news says it's 90% mortality, and Ebola kills everybody, and it's the hot zone, and et cetera, is really from the story of this initial Ebola outbreak, because it was so shocking and it was so unlike anything people had really ever seen. And this is in an area where hemorrhagic fever viruses are endemic. So it was really a big deal in terms of emerging viruses, and there have been many Ebola outbreaks since, of both Zaire Ebola virus, which is what this is, Sudan virus, which I just mentioned. There's another one that infects humans called Bundibugyo virus that's in Uganda, and these have sporadically occurred. So in 2013, actually, it was probably December 2013, that this 3-year-old boy was playing in this tree in the village of Meliandou, Guinea, where there was a colony of these Angolan free-tailed bats living in this tree. That boy ended up being the initial patient, patient 0 of the West African Ebola outbreak. And that also has further linked Ebola to bats, although unfortunately, when the people in the village found out that it might have been the bats, they burned the tree down, so nobody could actually look and see if the bats living in that tree had Ebola. But it was likely exposure to bats that initially caused it. Then the outbreak smoldered for months, like all the way basically until the summer of 2014. There was not this exponential growth that then later occurred, and I'll talk a little bit about why that was in a minute. But really, it kept spreading essentially due to poor healthcare infrastructure, lax border security, and a severely delayed response by global public health officials. So when we talk about West Africa, it really was the first Ebola epidemic. I mean, this is the first time that thousands of people have contracted it. Prior to this, the biggest outbreaks were a couple hundred people, not insignificant, especially considering the high mortality rate. But this was the first time that we really saw what would happen when Ebola got into a large urban mobile population. So this is West Africa. We actually didn't think that we would see Ebola in West Africa. So there's another species of Ebola virus now it's called Thai forest Ebola virus, but it was called Côte d'Ivoire Ebola virus, that emerged in Côte d'Ivoire in the mid-90s. And it appeared to kill chimpanzees. It killed over half the chimpanzees that lived in this forest, the Thai forest. And it infected one woman who was a veterinarian who was performing necropsies on these dead chimpanzees. So she became infected with it, but she didn't die. So it's not thought to be a major human pathogen. But that was, prior to this, the only time we'd ever seen any kind of Ebola at all within about 2,000 miles of this. We certainly did not expect to see Zaire Ebola virus, which is the big bad original Ebola virus, emerge from in West Africa. But that's what happens with these emerging diseases. They pop up out of nowhere and really surprise you, usually in a negative way. So this was really the perfect storm in West Africa. Liberia, Guinea, and Sierra Leone have all, in the last 50 years, recovered from these brutal, very bloody civil wars. There's a lot of mistrust of the government, unstable governments, very terrible public health infrastructure, and limited access to health care, very low per capita healthcare spending. Then on top of that, there was apathy and inaction from the global health community. I think people thought that the sort of tried and true Ebola methods were going to work, which is, traditional epidemiology, contact tracing, quarantine, isolation, and the outbreak will be over. But they didn't count on the fact that between these 3 countries, there was essentially no border security, and people frequently would go back and forth visiting their friends and relatives in other areas. On top of that, there's another hemorrhagic fever virus that's endemic in West Africa. It's called Lassa fever. There's also malaria, which mimics some of the early symptoms of Ebola virus as well. So when these Ebola patients started showing up in the hospitals where these people were very accustomed to see Lassa and malaria, they didn't initially think Ebola because, again, there was no Ebola in West Africa prior to this. So this really created a perfect situation in which Ebola was able to spread into these urban populations and really got out of control much more quickly, I think, than anybody thought it would. So that's all good. But how does Ebola actually cause disease? Because, I mean, you hear about the hot zone, and it's like, you know, weeping tears of blood and like this, you know, bleeding everywhere. And I reread The Hot Zone recently, and you know, that's why I became a virologist. I read it when I was in high school, I think. And I read it now as a virologist, and I thought, oh my God, like, I can't believe that this guy wrote this, because a lot of it is very, very heavily exaggerated about Ebola. So let me just tell you a little bit about Ebola virology. So like malaria, Ebola is a parasite. Unlike malaria, Ebola is a virus, so it's not actually even a single cell. And you can debate for hours with other virologists whether or not viruses are even alive, because viruses can't reproduce on their own. They're obligate parasites, and they require a host cell to reproduce. So they're much smaller. They have a very small genome. I think malaria has 60 genes or something like that, I think you said? Oh, thousands, okay. Yeah, 60 for the antigenic switching, yeah. Ebola has 7. So it is one long 19,000 nucleotide genome that encodes 7 genes. A huge chunk of it is the polymerase, which is the enzyme that replicates the RNA of the virus. And it does all of this crazy pathology with just this small amount of genetic material. So what happens is when you're exposed to Ebola, probably through a cut or through, you know, an injection site, close contact with vomit, blood, et cetera, from an Ebola patient, what happens is the virus is, you know, it's sort of like this thread structure. So it leans up against the cell, the outside of the cell. It sort of attaches loosely to a variety of different proteins that are on the cell surface, and then the cell takes it inside. And that part of the cell outside the nucleus but inside the membrane is called the cytoplasm. It goes into the cytoplasm where it starts expressing its viral genes. It starts making new virus. And then it basically takes the virus, puts it all together in a process called assembly, and secretes it out of the cell where it can go and infect a new cell. So that seems pretty simple and straightforward, and it's pretty much like what almost every other virus does. No, not everybody, I'm generalizing, but most viruses have, especially RNA viruses, have a replication cycle that's very similar to this. So why is Ebola different? Well, part of the thing is that Ebola infects very specific cell types, and it infects multiple cell types, and it has this effect where all of these cell types together are just dysfunctional enough to create this terrible systemic illness. So the first cell types that it infects are the monocytes and macrophages and dendritic cells. Now these cells are phagocytes. They're cells that run around gobbling up material. Dendritic cells, their job is really to take that material, go to the lymph nodes, and decide whether or not they're going to activate B and T cells to go out and start taking care of an infection, or whether it's a false alarm. Monocytes and macrophages have that role as well, but they also can stimulate inflammation very powerfully. So what happens is the virus infects these, it goes into the dendritic cells, they go back to the lymph nodes where they actually start killing off the B and T cells so that you can't mount an immune response. Simultaneously, they're producing more virus, so it gets into other dendritic cells that are in those lymph nodes and what causes what we call viremia, which is levels of virus in the blood. So the macrophages at the same time are infected, and they start dumping out these pro-inflammatory cytokines, which are molecules that basically tell the body when to, you know, get inflamed. So if like you get an injury, for example, it becomes red, it becomes painful, it becomes swollen. That's because of the local action of pro-inflammatory cytokines and chemokines. When it happens that it's happening indiscriminately and these cells are dumping huge levels of these molecules out into your bloodstream, you get a systemic inflammatory illness. And that's really how Ebola starts to get you. It causes what we call a cytokine storm, which is exactly what it sounds like. It's just a raging pro-inflammatory cytokine mix going through your bloodstream. After you get high viremia, the liver becomes infected. As we just heard, the liver filters your blood, and the Kupffer cells, which are basically liver macrophages, along with the actual hepatocytes, the cells that make up your liver, are all susceptible to infection with Ebola. The liver is also where a lot of your blood clotting factors are made. So as these cells become infected, they die. Your body can't produce new clotting factors. The cytokines circulating in your bloodstream cause what we call disseminated intravascular coagulation. This is when the inflammatory mix causes the circulating clotting factors to just spontaneously clot. They form all these little, they're called microthrombi. They're little microscopic, little almost like grains of sand, but they're made up of your clotting factors. When you have enough of these, basically you don't have any clotting factors in your blood that can clot anymore. Simultaneously, the last step in this process is that your vascular endothelial cells, the cells that line your blood vessels, become infected. They then will, there's a signaling process that occurs that causes them to separate from one another, and that causes your blood vessels to become leaky. So you've got no more clotting factors because they've all been made into microthrombi. Your hepatocytes are dying, so you can't make new clotting factors. And now your endothelial cells are messed up, and they're basically letting all the blood out of your blood vessels. So that is how Ebola hemorrhagic fever occurs. Now, that doesn't happen in every case, and there are many cases of lethal Ebola that never get to a point where, you know, there's tons of blood coming out, or else the hemorrhagic symptoms are more moderate. You know, bleeding gums, things like that, a petechial rash, and I'm about to show you a really gross picture of a petechial rash, but many petechial rashes are just, you know, a rash, essentially, on your skin. So it's not this sort of horror movie situation in every case. What may be more horrific, or just at least gross, is that in the West African epidemic, it seems that one of the symptoms that's most associated with mortality is the gastrointestinal illness. So there's a lot of vomiting, a lot of diarrhea. It doesn't have to be bloody. It's very efficient at transmitting the virus, and it's very efficient at killing people by dehydrating them. So Ebola is a very complex disease. This model of Ebola pathogenesis has basically been developed to show you will get Ebola through a small cut or a wound from its natural reservoirs, or an infected non-human primate infects the macrophages, and ultimately this leads to vascular leakage and these clots that drift through your bloodstream and basically make it so that you have completely defective coagulation system. There are many different symptoms of Ebola. As I mentioned, this can present in all different ways. My work in the Cates lab has been using a mouse model of genetic diversity to understand how different backgrounds, different genetic backgrounds will lead to different disease in the same animal. In my case, I use a mouse model. But what we know is that in different people, for probably genetic reasons, also environment, general health, any kind of comorbidities they may have, they have malaria or yellow fever or anything else, it can affect the way that Ebola presents. So there are many different symptoms of Ebola. The most common ones are headache and fever. However, now it's becoming more and more appreciated that gastrointestinal symptoms are also potentially a huge risk factor in terms of mortality. So I'm about to show some gross pictures, so just close your eyes. I'm not going to sing. That would actually be more horrifying than these pictures I'm about to show you. But if you don't want to see gross Ebola pictures, like, close your eyes now. But these are the most severe cases of Ebola in the clinic. This is a really, really horrible petechial rash where effectively the vascular leakage under the skin has caused these large blisters to form on this patient's arm. Many other patients, though, have these symptoms up top, commonly very red eyes, although I have not yet heard an account outside of the hot zone of people actually weeping tears of blood. But this is a very common effect. Also, as I mentioned before, bleeding gums, bleeding tissue, mucosal tissue in the mouth and nose is a fairly common presentation of the hemorrhagic disease. We knew from this outbreak, at least, that the risk of transmission is primarily limited to people who have very close physical contact with somebody who is actively sick, somebody who's viremic or a dead body. So, you know, there was a huge effort to control the West African outbreak by educating people, people on the ground in Liberia, in Guinea, in Sierra Leone, about how they could minimize their risk for getting Ebola. But you're not going to get on a plane, and you're not going to get it if you're R&B star Akon, who did a concert in the Democratic Republic of Congo. They also had an outbreak of Ebola at the same time as the 2014 outbreak in West Africa. And he was so concerned about getting Ebola that he manufactured this hamster wheel thing so that he could crowd surf at his concert without being exposed to a potential Ebola patient who I'm not sure why they would be at an Akon concert if they had Ebola. But you know, people were doing stuff like this to go to extreme lengths to avoid Ebola. And again, it's not really based on any hard data. It's mostly based on fear and alarmism. So can you get Ebola on a plane? That was the big question everybody wanted to know. I don't think anybody will say it's impossible. You certainly could if there was a big puddle of Ebola vomit on your seat, but I think, you know, it's pretty unlikely that you're just going to be sitting next to a person, or they're going to even be allowed to board a plane. And there have been instances of multiple Ebola patients who got on planes during the outbreak, including the gentleman who went from Liberia to Nigeria. He imported Ebola to Nigeria. Nobody sitting on the plane next to him got Ebola. It was only people who were in very close contact, meaning people shaking his hand, people sharing meals with him, sharing glasses, things like that. You're not going to get it most likely from getting it on a plane. So, you know, UNICEF, the World Health Organization, the CDC, NIH, all the partners who tackled Ebola during this really tried to get out the message that, especially in West Africa, that there are ways that we know that Ebola is spread, and we should focus on preventing those rather than freaking out about other possible ways that Ebola might make it over here to America and cause, you know, a pandemic. I would worry a lot more about flu shots. When you're talking about pandemic illnesses, Ebola is not that. So I think, you know, a really important message for any future emerging disease outbreak is, you know, slow down and try to figure out what the actual facts are, not what they say on cable news. I was invited to go on a couple of cable news shows, and I think I got kicked off of Fox News because I refused to tell people that they shouldn't fly or travel anywhere. Literally, Neil Cavuto cut like my segment in favor of the person who was like, we're all gonna die. So this is a map showing all the different Ebola outbreaks, and showing if we know how it was initially spilled over into human populations and then how it was transmitted. So in almost every case, this results from a spillover, either contact with a bat or non-human primate, but then it's person-to-person transmission. It's not like people are constantly hanging out with bats after there's an Ebola outbreak going on. So you know, it just goes to show that Ebola, for the most part, has been very sporadic. And up until West Africa, it has been largely controlled where it's occurred because of these more traditional epidemiological techniques, making sure that people who have been exposed are isolated until the incubation period passes, making sure that people are burying their loved ones in a way that respects their cultural traditions and their dignity, but also protects them from exposure. And this has been pretty effective, and ultimately this is how the West African outbreak was brought under control, but it just took a long time. So this is how we think that generally Ebola spillover and epidemic transmission occurs. It begins in the forest with exposure to a reservoir species. Sometimes it can be through direct contact with a bat, sometimes it's with a non-human primate who's been infected. Then it moves into these more rural communities. It affects people largely in the initial infected person's family, especially, as I mentioned, the women in many places in Central Africa. Women they play a central role in funeral rituals and also in healthcare. So there are traditional healers as well that usually are the first ones to be exposed. Then, in the case of the West African outbreak, it moved into urban towns and cities, got into the hospitals then, and that's when you know, it was really tragic, especially for Sierra Leone. They had a hospital where some of their leading researchers who studied Lassa fever all started treating Ebola patients, and you know, at least 5 or 6 of them died just treating the Ebola patients, you know, doing their jobs. And so as a result, you know, their precious intellectual resources were also depleted and lost to Ebola. So this is really how this cycle works. And there are a lot of opportunities for intervention from an epidemiologic standpoint. And I'm not even getting into the vaccines and drugs, although I will mention a bit about that briefly in a moment. So really, what did we learn from the West African epidemic? And we learned, first of all, that this was far larger than anything we'd ever seen with Ebola before. At this point, this was a couple weeks ago, the World Health Organization they consistently update their data on this. There are a few cases that have been flaring up in Guinea especially, so they constantly update it. I mean, 11,000 people have died. That's huge. Before this, the most that had died in any Ebola outbreak was a couple hundred. It's still nothing compared to the people who die every year from malaria, from other endemic emerging or re-emerging diseases. But this was the largest outbreak ever, and it really provided us with an opportunity to understand how Ebola will behave, or the kind of disease that it will produce in a larger, more diverse population of people. And one point I'd really like to make, though, is that this death toll could really have been prevented. So this is the graph of the number of Ebola cases in the 3 countries most affected in West Africa from April through October 2014. This arrow shows the time that people in America and in the West started caring about it. This was when Dr. Kent Brantley was airlifted from Guinea and brought to Emory Hospital and was, all over the news, and should we bring Ebola patients into the country? And I mean, if you're gonna bring them anywhere, send them to Emory. It's like a BSL-4 hospital. But it wasn't really until he got in the news that people even started paying attention to this. And you see that really happened right around the time that this went from a smoldering outbreak to a dramatic increase in the number of cases. And at that point, it was really difficult to put the horse back in the barn. We finally did it, but it could have been prevented. I think a lot of these deaths could have been prevented with more rapid action. And part of that is that there should have been more rapid action initially when the outbreak started, before it started getting into the cities, before here on the bottom, this was at the peak of the outbreak. You literally had dead bodies lying in the streets of Monrovia. And it's a terrible human tragedy. But even prior to the outbreak, there hasn't been action taken. And I think this is one of the major messages I want to drive home. It's really important, even if a disease is not the most popular or the most profitable, there's a broken market for it. We had 2 very effective Ebola vaccines that had been developed, tested extensively in non-human primates. One of them is now currently undergoing clinical trials in Liberia. These vaccines are so good. The one that we've worked with actually it produces sterilizing immunity. If you have this vaccine and then you get infected with Ebola, you will not get infected with Ebola. You can't detect virus. It's that good. Unfortunately, these were never moved forward beyond the point where they were tested in non-human primates because there was just no profit in it. There was nobody to fund that work. And same with ZMapp, the other antivirals that have been tried. There was no infrastructure to even provide supportive care, which now it's quite clear that by providing fluid support and things like that, you really can improve the clinical outcome for many of these Ebola patients. So just the absence of all of these things was really a way that we could have prevented this or the next one. And I really hope that the global public health community takes this as a very important lesson, that even though past Ebola outbreaks have been easily contained, and even though it doesn't affect a lot of people, that's no reason why you should be not lazy, but you shouldn't be proactive about handling it, and you also should not avoid funding riskier products, because it really could have saved a lot of lives. So I'm just going to finish up by talking about how we're going to study Ebola and other highly pathogenic viruses going forward. So we have a collaboration with NIAID, the National Institute for Allergy and Infectious Disease. It's an NIH facility in Hamilton, Montana, where they have a BSL-4 high containment lab. So we've done a lot of work with them to study Ebola. We've done some MARBURG work. Lassa and Lujo is another emerging hemorrhagic fever virus that's related to Lassa. We're doing some work now on 1918 flu, H5N1, H7N9 avian flu, and MERS coronavirus, which doesn't require a BSL-4 lab, but we do it with them anyways. Cynombre virus, which is a hantavirus that emerged in the 90s in New Mexico. Tick-borne encephalitis virus, which is not here, but it's endemic in Europe and Asia. And then, of course, the flavor of the year, Zika virus, which now everybody's talking about. So we can do this work. There's only a few of these labs in the country where we can work on highly pathogenic viruses, select agents, and BSL-4 is the classification where you have to wear these positive pressure suits like my colleague Atsu is doing here. He always says he looks like Buzz Lightyear. But it's very difficult to work, and it requires a lot of training, a lot of skill to do this type of work. But the reason why it is like this, and this is where I just shamelessly stole your idea to use a Gary Larson cartoon, is that I've never worked in a virology lab that didn't have this Center for Viral Pathology dropping, the petri dish out of the window and saying, uh-oh. Every single virology lab I've ever worked in has this cartoon hanging on some wall in the lab. The whole thing that's funny about it is that if this were to happen now, you could count on losing all your funding, your lab would shut down, and you'd probably never be allowed to work with anything pathogenic ever again. So that's why we have this mechanism in place, and especially after 9/11, there's been a huge emphasis on biodefense, so this actually cannot happen in the United States. If it did, it would be a huge problem. I think it's highly unlikely that an accident like that would ever happen. And we have this network of national biocontainment laboratories. There's only about 5 that can do BSL-4 work, so work with Ebola. There's a number of others that can do BSL-3 work, so that's work on MERS coronavirus, SARS coronavirus, influenza, things like that. So things that still require high containment but don't require the same level of containment that is necessary for working with Ebola. But we have this whole network of laboratories, and this is where this type of work is going to be occurring. As more and more open, they've just started another national laboratory in Boston. Well, they started a long time ago, but now it's finally going to go hot and be BSL-4. There will be all these different locations that you can study any emerging virus that comes out, and hopefully we can be really proactive about future viruses that emerge. Because I think that we're going to be seeing a lot more emerging viruses. I mean, before Ebola, there was chikungunya. Dengue is still a huge problem. Dengue affects probably 1 billion people in the world. Hawaii is currently in the midst of the worst dengue outbreak it's ever had in 40 years. There's a lot of these viruses that are coming, and with climate change, I think we can just count on more and more arboviruses in particular. So arboviruses are arthropod-borne viruses. They're transmitted by mosquitoes, ticks, and sandflies primarily. And as you can see, there's a lot of viruses here. Some of them you've probably heard of. Some of them, like, what the heck is Chiasanur forest disease virus, There's a lot of these viruses that are like Zika, where they've emerged. We know very little about them, and they could emerge again at any time and spread. Prior to Zika, chikungunya, which is also transmitted by Aedes aegypti mosquitoes, emerged. It also started in Africa. It spread like wildfire through the Caribbean and through the Americas. It causes a persistent arthritis, and in some cases, it's now believed that it can cause encephalitis too. Zika obviously has been linked to microcephaly. It's been linked to this inflammatory disease, Guillain-Barré syndrome, and who knows what else will emerge next and cause a whole new epidemic. So my final message is just that we need to be very mindful of the fact that this is not a problem that's going away. Emerging viruses are here to stay. This is a map showing where they think the risk will change for contracting dengue, which is, again, transmitted by Aedes aegypti mosquitoes. They are limited to tropical areas, and as you can see, as the climate warms, there's more and more areas within the United States and Western Europe that these mosquitoes can now live. So if you're living in, I guess, like Nebraska or New Mexico there, all of a sudden your risk is increased over 100% for contracting dengue virus where you live. And as I mentioned, there's a huge dengue outbreak occurring on the Big Island of Hawaii as we speak. So that's the message I'd like to leave you with. I would like to thank you all for inviting me here to speak. I'm sorry that I didn't sing, but trust me, you're better off for it. And please, anybody is more than welcome to email me at any point with questions. Thanks.
Okay, maybe we could take 2 questions.
Sure.
And then people could come up and speak to Dr. Rasmussen at the front if they'd like.
Absolutely.
Well, thank you. That was great. So my question to you is, you're making it sound like the message is if you have early identification, you can take very standard, well-defined epidemiological measures sounds great, but you put up a list of 100 very strange-looking viruses transmitted by common things. So like in the United States or Hawaii, for example, how would you and a whole bunch of other well-educated, smart, well-funded people find those 200 different viruses and figure out whether it's malaria or one of those?
Fortunately, it's pretty easy to distinguish malaria from a virus, so that's an easy one. But how would we distinguish, I guess, so a couple things. First of all, there have been tremendous advances in genomics technology that will allow us to identify a given organism or a given virus based on just a small blood sample. And actually in West Africa, some of my colleagues at Rocky Mountain Labs tested what's called a nanopore sequencing system, and this is literally a handheld sequencer that could diagnose people by fully sequencing the Ebola genome in a handheld device. So technologies like that will help us understand when there's a given outbreak of an emerging virus that we don't know what it is. It'll help us to understand what we're dealing with. If it's a completely novel virus that we've never seen before, hopefully it will at least have something in common with some other virus that we have seen before, so we might have an idea of at least what virus family it's in. But really the most important thing is to figure out which of these viruses is going to emerge. And there's also a ton of people who are doing predictive modeling, trying to predict, which of these viruses will go this year into the Caribbean, for example, like chikungunya and Zika have done, which are likely to emerge and continue to be a threat. Dengue is always a threat. But what you would do when you say, for example, it's a novel mosquito-borne emergent disease or re-emergent disease, I'd say, mosquito control techniques, insecticides, insect repellents, DEET. I'm always preaching the gospel of DEET to everybody. Nothingbutnet.com. Although we don't need that kind of help here, but certainly dengue is a huge problem in many of the areas that malaria is too. So working on insecticides, mosquito control, insect control is a very important way. For viruses like Ebola that aren't transmitted by an insect vector, you need to understand the ecology of these viruses, and unfortunately we just don't know that much about it. But you can control person-to-person spread. And for most viruses, even if we don't know that much about the virus, we still can do that contact tracing, that traditional basic epidemiology, and that really can help too. I think the problem in West Africa was not so much that wasn't done, it just there wasn't as rapid of a response, and there wasn't an understanding of how widespread the problem was. People thought it was limited to one area. They weren't accounting for the mobility of the people in the affected areas. They weren't accounting for the fact that the rules would change basically when it got into an urban setting. So I think taking a lot of those lessons and just not thinking that, oh, it's another emerging virus, like we're gonna control it just like we've done every other one, but being proactive and getting in there and really trying to stop the spread before something becomes a huge epidemic, however we can, is gonna be critical.
What are the major molecular factors that allow you to distinguish Marburg from Ebola?
Clinically, there aren't that many differences, to tell you the truth. I mean, Marburg can cause a raging hemorrhagic fever like the worst cases of Ebola, but both Ebola and Marburg can also cause more mild cases, and they both cause very similar disease presentations. Historically, they used to use antibodies to distinguish between them. Now they use polymerase chain reaction, or PCR, and increasingly people will be using sequencing as well. So not only can you define this is Marburg, this is this species of Marburg, or this is this species of Ebola, but you can actually even determine what strain it is. And that has been done in the most recent Ebola outbreak as well. The so-called Makona variant is what the West African Ebola is, and while the outbreak was still occurring, there were people publishing papers on the sequence, sequencing multiple viruses and isolates from multiple people. So we really have an understanding on a molecular level of what these viruses are doing more rapidly than we have ever in the past in the history of epidemiology. So those methods going forward, I think, will be integral to our epidemiology efforts.
All right. Thank you very, very much. That was great.